The MethylFlash Global DNA Hydroxymethylation ELISA Easy Kit is a simple and convenient way to rapidly screen genome-wide levels of the 5-HMC modification in your DNA samples. The assay with this kit can be completed in just 2 hours, and results are comparable to MSLC analysis, reflecting true status of sample DNA hydroxymethylation. In the assay, DNA strongly binds to specifically treated microplate wells. The hydroxymethylated fraction is detected using a highly specific antibody and then quantified colorimetrically by measuring the absorbance in a plate reader. The kit contains all essential components needed to complete the assay. Begin by preparing the standard curve. Combine the positive and negative control DNA in microfuge tubes to generate various 5-HMC concentration points as outlined in the user guide. Arrange the required number of well strips in the plate frame. Add binding solution to all wells. Then, load the prepared standards and negative control into duplicate wells to establish the standard curve. Next, load your DNA samples into duplicate wells arranged in a similar manner as the controls. Gently shake the plate to mix. Cover the plate with the provided adhesive film. Place in a 37°C incubator to allow DNA binding to the wells. During the last few minutes of incubation, prepare the 5-HMC detection complex solution by diluting the 5-HMC antibody, followed by the signal indicator and enhancer solution, as described in the manual. Retrieve the plate from the incubator. Remove the binding solution and wash the wells with diluted wash buffer. Add the 5-HMC detection complex solution to all wells. Cover the plate and incubate at room temperature. Once incubation is complete, wash the wells again to remove unbound detection complexes. Add developer solution to all wells and monitor the reaction as a blue color develops. When the desired color intensity is reached, add stop solution. The color will change from blue to yellow, and the plate is now ready for measurement. Read the absorbance at the recommended wavelength. Plot a standard curve using the OD values of the control DNA concentration points and calculate the percentage of 5-HMC in your samples. With the MethylFlash Global DNA Hydroxymethylation ELISA Easy Kit, you can achieve fast, sensitive, and reliable quantification of 5-HMC. Please visit epigenetic.com or contact your local distributor for more information.